everyone. So I thought I'd do a series of short videos just to show you some of the basic aseptic techniques for tissue culture and actually how to do the practical. Starting off with these gloves, what you need to do is make sure that your cuffs are well below and the gloves extend over these cuffs. That's the first point. Okay? If you've got that ready, next thing you do is take a piece of glue roll. Swish it with 70% ethanol. The next thing you do is swish the entire contents of the cabinet with 70% ethanol all the way across. And last on the glue roll, and now start cleaning it, starting from the back and then working your way forward. Discard this in the white autoclave bin. The next thing you have to remember when you're doing this is that anything that goes in the tissue culture hood has to be squished or squirted with 70% ethanol, so including tip boxes. Now you can see I have already kept some which have been squirted with 70% ethanol. It also includes your pipettes, again squirted with 70% ethanol. Keep it over there. And lastly, 96 well plate, just squirt and keep it over there. The next thing you need to know is how to use a pipette. Now this is a sterile pipette that's used only in the tissue culture lab. They're individually wrapped and aseptic. There's a technique on using this. So what I would suggest is first make sure you rip the plastic off. Don't take the whole thing off, just a small bit at the top. Put it inside your pipette and take the sleeve out, keeping it carefully on the left hand side. If you're right handed, make sure all the clean stuff is on the right side and all the discards etc. goes on the left side. So keep that sleeve aside. Then you need to make sure you are able to take the lid off from the universal like so, using one finger. Drop the liquid to how much you want. Replace the lid. Keeping the lid held in your same hand, discard the liquid in your container. When you're finished, tighten the lid back and you can put it back in the sleeve. Now one thing to watch out for here is that the tip of this pipette should not touch any other surface. If you do, that's when you'll get bacterial or any other contamination problems. So if anything has touched your pipette, the best thing to do is to get rid of it. Okay. Now I'll show you what you need to do for day one of the practical, the Monday when you're adding the drugs to your cells. The first thing when you start, just make sure you squish a piece of glue roll and keep it on the side over here. Make sure you squirt your hands, gloves, completely. What you should find kept in front for you are the tips and the microfuge tubes. Now what you see here are microfuge tubes. You need 4 of these. And label each tube either 0 or drug A, drug B, or drug C. The next step is dilutions. So now I'm going to show you how to do the dilutions. We've discussed this in class already. We've worked out the calculations that you need to go from a stock solution to the final concentration of drug that you require. So previously we saw how you labeled your 4 microfuge tubes. You should have stock solutions of your drugs supplied to you. It would be either in the incubator or it would be out here in the cabinet itself provided for you. All you need to do now is take your stock solution and make it up in your drugs with the diluted final concentration that you want in these tubes. Okay, let's show. You start off with the volume of fresh medium that you require. For example, for drug A, as we saw in class, you have to add 100 microliters of stock A to 900 microliters of fresh medium, and you add that to your tube A. Okay, so I'll take 100 microliters of the drug stock solution. Add it to your stock solution or final solution A. Discard the tip in the beaker labeled waste. Now to this, if you remember again from the calculations, I need to add about 900 microliters of medium to make it to a final volume of 1 ml. So here I am taking 900 microliters. There should be a petri dish labeled fresh medium provided for you. Take your 900 microliters and add it to tube A. And there you have it. Give it a mix and your final solution of drug A is ready. Discard your tip in the waste and do the same for drugs B and drug C. Remember, drug 0 is medium alone, so all you need to do is take 1 ml of medium and add that to your tube 0.